Hello, curious minds, and welcome back to another episode of the HEAS Podcast. I'm your host, Victoria, and I'm happy to bring you another episode full of science knowledge and two exciting new guests. Our first guest is Thibaut Devièse, who's a chemist working in archaeological sciences. His journey began when he developed an interest in archaeology early on during his high school years, but then received valuable advice to go into chemistry first and then with the obtained knowledge move into the field of archaeology. And here he is today, developing, improving, and applying a range of fascinating methods to ancient bones and other materials. Tibo is telling us about redating what was thought to be the latest Neanderthal remains from Vindija cave in Croatia, and the very surprising results he obtained from that. He talks about working on an unusual mass grave in the Roman catacombs during his PhD, and his very cool and super diverse hobbies. Our next guest is the lovely Chiara Barbieri, who's a geneticist exploring the human past using genetic and linguistic variation. This was such an interesting conversation for me personally because as our conversation moves on, you can clearly tell that I needed some education on this topic, which is good. I mean, I'm here to learn. And it was really cool to hear from Chiara how old some of these language families actually are when they emerged and how you can match genetic diversity with linguistic variation. We also discussed the very special case of the country Hungary and its unique language, which is not related to any of its neighboring countries' languages. Then we moved on to talk about the Inca Empire and the spread of the Quechua language and just so much more. So stay tuned and enjoy this really engaging conversation that I had with Chiara. I really hope you're going to like this episode and are able to take away some cool science facts. As always, stay curious, stay engaged, and welcome to the HEAS Podcast. Thibaut, welcome to the HEAS Podcast.
Thank you. Thank you for having me today.
No, we're so lucky to have you. You previously gave a talk at our lecture HEAS seminar series, and Tom has previously today introduced you as an archaeological chemist. That sounds really interesting. So could you please tell us more about yourself and what an archaeological chemist does?
Right. Yes, I have a training in chemistry. My degree and even a PhD is in analytical chemistry. And I did study chemistry knowing I wanted to apply that to archaeological questions, not a specific period of time or not a specific country. But I did study chemistry, and now I'm using it to understand better our past.
And as Tom also mentioned, somebody gave you actually the tip to not directly dive and study archaeology, but to go into chemistry first. Is that correct?
Yeah, that's correct. When I was at high school, I went to visit museums and about archaeology, and someone told me, if you one way to study the past could be through chemical analysis. So you may want to do a chemistry degree, and if there isn't a job in archaeology, you can always use your chemistry degree for something else. And so I followed that advice, did my degrees in chemistry, and did all the internships and even the PhD in museum environments, and I continue to do so today.
That was such a good advice because it's actually true. And if you are interested in archaeology, you wouldn't think about that probably in the beginning. And it's true, you could apply your knowledge to so many other fields. But as a prerequisite, I guess you have to be good in chemistry.
That came from a conversation about what I did like at school, and I said yes, I did like chemistry, and that's where I say you may want to do chemistry and then apply it to archaeology.
Yeah.
And now it's quite well known by many people that we can do chemical, physical, biological analysis on artifacts from the past, but it's, let's say, 15, 20 years ago, people knew about radiocarbon dating, obviously, but not necessarily all the other things we can do on past.
It was probably
archaeological
really good
material.
advice. Yeah. So how does your background in chemistry now play a role in your archaeological research?
I would say it plays a very central role in my research, because I spend quite a consequent amount of my time looking at methods in other fields and try to see if we could apply them to archaeological material and what it would help us to discover. And there are some aspects on reducing the sample size as well, because we work on archaeological material. This is precious. If we can reduce the size of the sample to do our analysis, that's great, because that means we preserve better our cultural heritage. So that it's about developing new methods to be able to answer questions we still can't answer, and also about optimizing the methods in the lab, so we are as little invasive as possible to preserve the material.
So one of the huge methods used in archaeological sciences is radiocarbon dating, and you've talked about that in your talk a lot, also about improving that method. So radiocarbon dating is a term that many people may have heard, but might not fully understand. And I'm also always happy to learn more, because Tom also has his radiocarbon dating lab. And I have to say, I still need a tour of that lab. So could you explain how that works and why it's important?
We use radiocarbon dating to give an age to things, to archaeological artifacts. And that method works with material up to 50,000 years old. And let's take the example of bones. This is the material that's most commonly dated in radiocarbon labs. So the bone contains collagen, we call the organic fraction of the bone, and that collagen contains carbon. And this atom of carbon, that's what we are interested in. And in carbon, you have 3 isotopes, carbon 12, 13, and 14.
So versions of it?
Electronically, they are slightly different, if I can say that way. And so 2 of them are stable. So over time, they will not degrade. So the amount of carbon 12 and 13 that was in the bone 50,000 years ago will still be the same today. The third one, the carbon 14, is unstable, is, we could say, radioactive. And I would say that the carbon 14 will degrade over time. And by measuring the ratio between the stable isotopes and the carbon 14, we are able to give an age to the artifacts.
Okay, so
maybe a bit too complex still in terms of explanation.
Let's see if I got it. So you have carbon 14, and you basically measure the degradation of C14 over time. And as we have a half time of C14,
yeah.
From there, then you can say half time is, I think, 5,730. Oh, it's been a while. So 5,730. And from that, then you can measure in your bone sample. You can see how much C14 is still left. And then from there, calculate back the date.
Exactly.
So why is it limited to 50,000 years of age?
So the reason we can't really go over 50,000 is because the amount of C14 will be so small that the instrument we have wouldn't be able to detect it, and actually at some point there is no C14 left, because all the C14 that was initially present in the specimen we tried to
will be gone.
Has gone. Yes.
Okay, so could you walk us through, because you've also mentioned the destructiveness and invasiveness of these methods, could you walk us through the process? So how destructive is radiocarbon dating, and how much bone material is needed for an analysis?
This is always a tricky question to answer.
I'm sorry.
Because what we are interested in is the collagen fraction of the bone, and for modern or well-preserved bone, it would be around 20%, but very often that collagen will degrade over time. So the amount of bone we need will depend on collagen preservation. I would say it's around 400 milligrams of bone powder. That's what usually we start with, but it also depends on the treatment you want to apply to the bone after. Are you going to date the collagen directly, or are you going to date a fraction of the collagen? And obviously, more selective you are, more material you need to take initially.
Yeah, that makes sense. And one of the main limitations, as we said, is the time that you can obtain. So you cannot go beyond 50,000. Another big issue is contamination. Could you tell us why that's a problem and how the dates are shifting when you introduce just a tiny bit of modern contamination?
So the contamination we have in the objects, the artifacts we try to date, usually has a different age. Most of the time, the contamination will be something that came after, so it will have a younger age. So if we extract the carbon from our sample, the endogenous carbon, there will be endogenous carbon, but there will be also more recent carbon. And so when we measure the isotopes of these two lots of carbon, we will get an age that will be younger than what we would have expected to find out.
You had a slide which was really interesting where you said if you introduce 1% of contamination, the older the sample is, then the more that...
As we just said before, over time, carbon-14 will, the amount of, the quantity of carbon-14 in the specimen will decrease. So 1% difference for something that contains a lot of C14 because it's quite recent, it's not a big issue. 1% modern contamination for something that's very old will have a big impact, because the older the sample is, less C14 there is, and any modern substance will have a lot of C14.
Talking about modern contamination and how it's introduced, you've told a story about a purported Neanderthal scapula. I think it was, right?
Yes.
Please correct me if I'm wrong, always. I'm happy.
That's correct.
And you found quite some interesting and maybe unexpected results in that. Do you want to tell us that story?
Yes. So that scapula is one of the specimens we analyzed from the archaeological site of Spee in Belgium, and when we asked our colleagues at the Max Planck in Leipzig to if they were interested in sequencing the DNA for that scapula, Neanderthal scapula, because it had never been done. So they did the extraction. Unfortunately, there wasn't any Neanderthal DNA preserved in that specimen because DNA can degrade over time. But what they found is modern human DNA and bovid DNA. So
bovid and like cattle.
Cattle, yes. And so what we understood, thanks to their work, and the anomaly we had with a radiocarbon date, is that this specimen has been preserved with a glue made from animal parts. And
the other conclusion could have been that it was a minotaur. I love that.
That's
I could tell Tom loved that slide.
It's always interesting in the middle of a very scientific presentation to put a slide that makes everyone laugh.
That's the best. Go on. Having heard all of that, and that you're focused on the one hand, developing new methods while also improving the already established methods, looking towards the future, how do you see the field of radiocarbon dating evolving?
I would add to your question that I like to develop new method or improve the method that already exist, but also apply them to very interesting archaeological questions. We have been talking a lot about methodological development, but it's not methodological development per se. It's with the idea of applying it to important archaeological questions. So coming back to your question about the future of radiocarbon dating, I would say more and more intercomparison between laboratories, because we all agree that it's important that we are using methods that are comparable, because if the methods are not comparable, that means that we can't put together data produced by different laboratories. And if for more chemistry-focused, chemistry-focused response, I would say we will see more and more compound-specific dating, because we now realize, thanks to all the methods we have in the lab, that it's very difficult to clean a material, but if we have the resources, we know how to select specific molecules for dating, and I believe this is what we will see more and more in the radiocarbon dating field.
Which then also goes hand in hand with a reduction in the contamination if you use compound-specific dating, right?
Yeah. So that will allow us to increase the quality of the data that's globally produced by all the radiocarbon facilities around the world.
Of course, if then this is set as a new standard and everybody's using it, it makes things easier to compare and throw everything into one study. And also, what you've done is applying these new methods and then comparing these dates that you obtained from previously dated material. Let's talk a bit about Vindija cave, because it's 2024. Last summer, I was so lucky to be part of the ongoing, or let's say the latest excavation at the site.
Nice.
It was such an honor to be part of that. And you've actually dated the Neanderthal samples from that cave that have previously been discovered. And would you like to walk us through what surprising results you obtained from that?
I like to use in presentation the case of Vindija cave, because we see the different generation of sample pretreatment and the impact on the ages we are obtaining working on the exact same specimen. So the dating of the Neanderthals from Vindija started many years before I even started working on radiocarbon dating. And what we have seen is with the development of the new pretreatment, the ages on the Neanderthal specimen has been going back and back and back, and I think it's about 10,000 years difference for the very same specimen between the first radiocarbon ages that were measured on collagen and the one we obtained on one specific amino acid, and obviously a 10,000 year difference for a single specimen can totally change the story of the site.
And it did. So that means that they got older, about 10,000 years older. And which implications does that then have on the story?
Based on the first dates that were obtained on the Neanderthals from Vindija, it appeared that this site may have been a refugium, so for the very latest populations of Neanderthals in Europe. But pushing back by 10,000 years the dating of this Neanderthal totally changed our interpretation of the site, and this wasn't anymore a refugium for late Neanderthal populations.
So the first dates were around 32,000 years ago, and that would imply that the Neanderthals would also have overlapped and probably interacted a lot with anatomically modern humans who were already present in Croatia at that time.
Correct. That's totally correct.
So, of course, while obtaining more resolution on the dating results, on the other hand, the story got a little, how can I say that? It would have been really interesting if these were really the youngest Neanderthals, but yeah, I mean, I guess that's science.
That's science, and there will be somewhere in the world the place where we will find at some point hopefully the latest Neanderthals. So the game is not over.
Okay, and with that, let's jump into our follow-up question section. So there's one question saying, which challenges have you faced during your career and how did you overcome them? Was it always smooth?
It's never, no, I don't think careers in academia are that smooth. One of the biggest challenges in academia is actually getting the funding to secure your own position. So I would say this has been probably the biggest challenge for quite a few years, applying for many grants and looking for permanent positions. So I don't know if that's really what you were expecting as a response, but to be honest, this is the main challenge is to get stability.
Thanks for saying that, because I think a lot of people are struggling with that, and it's not easy. We just had tenure track talks today, interviews, and it's really hard and super competitive at that stage to get a permanent position. So, what was your favorite research finding during your career?
I would probably go back to what I did during my PhD.
Yeah,
we are going to change totally period of time, because we are going to the Roman period, and we are going to Rome inside the catacombs.
Oh.
And I actually got one day a phone call from one of the archaeologists I'd been working with, because as a chemist during my summer holiday I was going on excavation, and this paleoanthropologist called me and say, look, I know you are doing your PhD in Pisa, and I have one of my colleagues excavating a new area in the catacombs in Rome, and they don't know what's going on here. There seems to be many people buried together. It's very unusual for the Roman catacombs. Could you go there and see if that could be interesting for you, if you could help with your chemistry to understand what's going on there? And I went there, went down inside the catacomb, and the archaeologist started to show me all these skeletons lined up. And she said it's very weird, because we are in a catacomb, but it seems that there is a lot of soil in the catacomb, and sometimes the soil is very red. And she did show me a little bag with some red powder, and when I looked at it, I said, this is not sediment, this is actually crushed resin. And it was very, very exciting. I told her, before doing any analysis, I'm sure this is a resinous substance, because I did work a lot on natural resins. And what we discovered, to cut the story short, is that they did some embalming of the bodies inside the catacombs. So they plastered the body with gypsum, and with inclusions of different resins. They used frankincense coming from Yemen, sandarach that was coming from North Africa, and the most exceptional result, and that red sediment was actually crushed amber from the Baltic.
Oh, wow.
And for that period of time, that was an extremely expensive substance. So what looked like a mass grave actually ended up being a site where many people had been buried with very, very special treatment, because we had individual bodies were plastered with gypsum, but also with very expensive resins coming from 3 different locations. So that's probably one of the most exciting.
That is exciting.
Results of.
And did you find
career.
more out about the people, like the context, as to like kinship analyses? Who were these people? Why did they get the special treatment?
Why did they get the special treatment? We don't know, and they even didn't represent. So they were not like only warriors. We had male, female, we had adult, we had children. So we think it's more of an epidemic that killed them. But it's quite interesting to know that if they died because of an epidemic, they still spent some time to do. I imagine quite a lot of time to do individual treatment to these bodies. So that we don't have all the answers.
Of course, yeah. But that's such a great and interesting topic. I would have loved to receive a call like that during my PhD. So if anyone, just call me. I'm up. Well, that's really cool. I'm so glad I asked that question. And it's so different from what we've talked about before. So
Indeed.
Really, really nice.
That's sort of what Tom said in the introduction. I had different lives, and I think the research you do at the university in a radiocarbon facility is very different from what you can do like in a museum.
Yeah, definitely. You could live different lives still. You could be like, I'm fed up with that. Let's move on. It's really cool how many options you have with that.
I have to say, again, that's why having a chemistry background is very helpful for that. It allows me to switch from one period to another one. And there are many questions for which chemistry can help, like understanding funeral rituals. That's what I did during the PhD. Or colorant on textile, figuring out what were the plant material used to dye textiles in the past. That's what I did quite a lot at the British Museum, and then doing radiocarbon dating on Paleolithic bones. So what's the common point between all this research, super exciting research topics, is the use of analytical chemistry.
What a cool career. I also have a friend who's a chemist, and he's now actually working at, or has been working for a while at our department, and I just consulted him for something that I just do not understand and I need input. So yeah, he's also very smart, and definitely chemistry is useful.
Don't hesitate to give me a call if sometimes you have questions and
Thank
you
you.
think
I might
help.
actually.
Don't hesitate. I'm
Thank
always
you so
happy
much.
to answer questions if I can.
That's very kind. Now let's move on into the rapid fire questions. Just answer spontaneously, okay?
I will try my best.
3 words to describe yourself.
Passionate, curious, and exigent.
Nice. I guess we can skip the second one because it says favorite subject at school. Maybe
difficult. There were chemistry, but also
Can you
history.
Oh, wow. Yeah, that kind of paints the picture of what you were going to... Yeah. What did you want to be when you grew up as a child? We know what you wanted to be probably in high school already a bit, but let's say as a child.
I had the period I wanted to be a doctor.
I could see you as a doctor.
And then I moved to archaeology.
To the dead.
Yeah, it's too late for them.
Last book you read?
I would actually say I haven't read a book for a very long time, because that's not what I find the most relaxing, and I prefer to practice music.
Oh, that's a good answer. What do you play?
Oboe. Do you know oboe?
Oh my God, I'm embarrassed to say no. What is it?
It's quite similar to clarinet in the shape of the instrument.
So my next question was going to be, do you have any unexpected hobbies? But that could be one of them. Anything else you want to share with us?
I think I gave you the one that usually surprises people, because they see me as a scientist talking about chemistry, and when I tell them that I actually play classical music, that surprises them usually.
That's what we want to know here in this podcast. You know, that's exactly the answer I wanted to have. Homebody or adventure? So would you rather, let's say you have a day off, would you stay at home or hike up a mountain? Or
adventure.
Yeah?
Traveling.
Yeah. Winter or summer?
Summer.
So rather a beach vacation than a mountain
Sailing.
Sailing? You keep on surprising me here. So you're a sailor as well. Amazing.
And diving also. It's not that fun to go diving during winter when the sea is very cold, but during the summer to go diving is much more pleasant.
True. I also did go diving in an Austrian lake, and I mean, it's nice, but it's extremely cold. I didn't want to wear a dry suit, so I kept my wetsuit on. Bad
idea.
Choice. If you could travel to any time period, where and when would it be?
That's funny. I actually was talking with someone recently and asked exactly the same question.
Oh, look.
And for me, the response would have been medieval time.
Why is
because
that?
many discoveries were done during that period. People traveling to country, bringing things from faraway countries. I think it was an exciting time in terms of art and expeditions, etc., etc. So
definitely. Now moving on to the last segment, where each guest that we have leaves a question for an upcoming guest. And I like that question because it kind of we're coming full circle to one of my first questions, where I asked you about why you became a chemist. So the question is, what is the best advice you've been given?
Very interesting question. And you're right. I would probably go back to the beginning of this conversation we had and say that the best advice I received was from that person who said, there are not so many jobs in archaeology, and if you like science, like chemistry or physics, maybe start with a degree like that. And if you are lucky and if you remain passionate about archaeology, you may end up using your chemistry knowledge in the field. But if you don't find a job in archaeology, you can use your chemistry for many other things. And I think that advice probably shaped my career. So I would say it's one of the best advice I received. So my advice to people would be never be shy to ask for advice about career, what to study, and then make your own decision. Don't let other people make a decision for you. I mean, I had several times students asking me, should I do a PhD after my master degree? And my only answer is, no one should tell you the answer to that question. You are the only one who can answer that question. You can ask for advice about what would that bring me to have a PhD in addition to my master degree, but should I spend 3 years minimum working on a topic? You are the only one who should answer that question. So go for don't be shy, ask for advice, but make your own choices, the one you think are the best for you.
Thanks for sharing that. I think that's also really, really good advice. To wrap this up, I would like to ask you, is there anything you feel like I didn't ask you or anything you would like to add?
I think we covered quite few things, from
we covered a broad spectrum.
The very scientific aspects of the seminar to knowing that I play oboe and I like to go sailing. So I think we covered quite few things.
It was such a good conversation. And now to end this, would you like to provide your social media handles, or where can people find you online?
So the best way to find me is probably on Twitter under Thibaut Devièse, and they should be able to find me easily there, and they can contact me, and I am always happy to answer questions when I can.
Thibaut, thank you so much for sitting down with us for the HEAS podcast. It was a real pleasure.
Thank you for inviting me, and that was a first experience for me for this kind of yes. Great. This kind of interview.
Hi, Chiara, and welcome to the HEAS podcast.
Hi.
To start off, could you please introduce yourself and also tell us a bit about your work?
Sure. My name is Chiara Barbieri. I'm a geneticist, and I work comparing human genetic history and linguistic history, understanding diversity of human groups today.
I think the interesting part for us here is that your work involves an intersection of linguistics and genetics. Could you explain how you use genetics, which is something biological, to trace language history, which is something more on the cultural side of things?
It starts with the concept that we can analyze the genetic history of people, track back their demographies, but ultimately genes alone do not speak. So to interpret what these results mean in terms of actual human history and trying to link this history with the identity of the people, we need to use some external evidence. It can come from archaeology, it can come from direct historical sources sometimes. And what I've been doing in my research is to put the linguistics in the mix. Mm-hmm. I think the use of languages is a way to reconstruct history in a way, and also try to connect group identity. So understanding the trajectories of different human groups, also thinking of the languages that they speak or they used to speak.
So what do you need? What kind of genetics are involved? Do you use ancient data and also modern? And how do you link that to the language?
So I mostly work with modern DNA, so with the
Mhm.
genetics of the people living today, of people that you can go have a chat with, ask them what is their history, where their ancestors used to live, which language do they speak today, or which language did their grandparents speak in the past. Mostly with, again, living groups. But sometimes I also use ancient DNA to anchor the genetic trajectories and these ancestries in the past in a specific region in time. We can say that the archaeological remains are even more difficult to connect with linguistic identifiers unless we work with people who had inscriptions in their own original languages buried in the same place with the human remains. That would be a one-to-one association. Otherwise, it's very tentative to link languages in the past.
Let's take the Neolithic maybe as an example, where there was a huge influx of new people. Do you think they brought the language in with them, like they also did with their domesticated cattle and everything and like seeds?
If we're thinking of the Neolithic in Europe, we are thinking of these large migrations of people carrying their domesticates with them, their new technologies, going towards the West in what is Europe today. Surely they were coming with their own cultural package, including a language. We don't fully know which language they were speaking at the time. There are contrasting hypotheses. Some associate these early migration and expansions into Europe to the origin of the Indo-European language family, which is the largest language family spoken today. We are speaking about languages that are related to each other in a family. Some people would then suggest that the larger migrations of the Bronze Age from the steppe have been the ones in a more recent time scale introducing the Indo-European languages in Europe. We don't have a full answer to this, but part of the game is understanding that a large migration, a large demographic effect might be the signature to explain the diffusion of a large language family.
It's really interesting that you need so many layers of information to kind of get some idea of which language it could have been that they spoke or which language that they brought in. And as you said, it's not even completely sure at this point even. So I think it's like a huge project to really piece everything together and then get some hints at the language that they spoke. So it's not really that nothing that you can find, nothing that you can touch, nothing that people really left behind. Only if you have like a piece of written, whatever, in written form, which probably is really rare, as I can imagine. But you have a really great project, which you called Gelato, Genes and Languages Together, where you're investigating the correlations between genetic and linguistic data. You've mentioned that in your talk today. Could you tell us more about that here as well?
Sure. The idea of gelato comes from conversations with linguists mostly. So if you ask me why is there so much linguistics in my work, it also can be explained by the fact that I've been spending time with linguists. So just chatting and having friends as linguists. And the questions they have is like, okay, maybe if we know more about the demography of the people in the past, this can inform us even when we are trying to understand some of these linguistic trajectories. But we need something to sit with us and come together with a plan, with a genetic diversity, and something that can be matched with the linguistic diversity that we have. So with this panel of genetic diversity, I've been selecting population genetic data available that could represent, in part, the history of some languages in the world, producing something that matches with existing large databases of linguistic diversity, quantitative data that has been put together and curated in particular in the past years. So we are now in a position where we can test things with linguistic data, linguistic features mapped for different languages of the world in search for patterns, in search for historical element and connections that we might have missed before. And we want to compare with the data of relatedness and history that we can reconstruct with genetics.
That sounds like a huge project in itself. My question would be, could you see a link between genetic relatedness and also language relatedness? Does that go together?
We've been assuming sometimes the languages and genes would have to go together because people move and again, they carry languages, genes, their cultural packages with them. So this sometimes has been like a baseline assumptions, especially for biologists. I've been analyzing this database, thinking of this in mind and thinking that I don't want to be biased and try to push for this solution to the question. But looking at the data from many angles, it comes out that very often we do see matches in this sense that are not just by chance, are not just because you tend to be genetically related to your neighbors and linguistically related to your neighbors, because they would be like the normal patterns of distributional diversity. But even across discontinuities, migrations, and movements of contacts, we see barriers that are genetic and linguistic. We see contact that is genetic and linguistics. And in different moments, very often, these histories match.
That is truly interesting that there is a match that you can discover, but if you think about how language and culture sometimes can rapidly change, even within a generation, as you mentioned, the grandparents and the language they spoke, it can also be just if you think about you're moving countries, your grandparents moved countries, and then they adapted to the language. And sometimes, especially a few generations back, they wouldn't even teach their children their native language. So it's really fascinating to me that you can still see to such a degree the match of culture and biology in the linguistics.
Yes, these are general patterns that we reconstruct for some cases where we do have data and we understand the process going on. But in your comment, there is actually a lot to unpack, if I must say, and it reveals how much language is connected to our sense of identity. And it reminds us that we are touching on topics that are very delicate. For example, even the fact that many people, like me, would say my grandparents used to speak a language, and then they shifted to a more common language, a language that is associated to a different status or privilege, is telling us a story of discrimination, where something that was connected to a certain identity in the past was pushed away and forgotten because it has been maybe discriminated. We've seen this all over the world, and it's a classic story that we are telling also when we analyze these genetic and linguistic histories. So this is another element that accompanies this part of the story. We also have these big shifts and migrations that happen today. In some cases, migrating people and contact with different languages produces some changes that are very interesting to study, because it's like studying evolution in action. Okay, maybe this is how new languages are born. So sometimes focusing on these even extreme contacts with different languages, we see what people do in a couple of generations, incorporating different cultural elements.
So you're basically studying the evolution of language.
This is a very big term that includes many things from the language faculty. Why do we have a language at all? I focus on this layer, which is the languages and the linguistic diversity of today.
As I'm listening to you, I'm realizing that it's a very complex topic, and it requires so many layers of information to really get a picture about what could have happened in the past. Even now, as you're saying, I mean, languages are constantly evolving. Talking a bit more about this correlation between the genetic and the language history, I'm sure that there must be a couple of mismatches where the genetics might be similar. Let's take a simple example. I think we as Austrians here, we're all aware of this specific case in Hungary where we're really close to that country, but genetically probably also. But if you look at the language, it's so different and it has nothing to do and is not related to the language of its neighboring countries. Could you explain that?
Yes, this is a classic case that we have in Europe of a mismatch with a group of people who speak a language that is very different from the language of their neighbors. Why do these things happen? It's not a trivial question to ask. We know that there has been a migration in the ancient time of the Magyars from Inner Asia that brought this language that belongs to the Uralic language family. This was a language of the elite, of the dominant elite. We know that ancient DNA is even showing us that the genetic profile of this migrant was indeed matching the profile of these Inner Asia regions. But with the time, this genetic signature got diluted and we don't really see it anymore, but the language stays because it has been associated to identity value, to a culture of a region, and it persists in Hungary today.
So you're saying that this language was associated with status and identity. These are two factors that are so persistent and so important that it's still to this day is so different from all the other languages around. Thanks for answering that question. I was always wondering.
Isn't it fascinating? Sometimes you travel to a place and suddenly all of your references are gone because you don't recognize even the words for numbers that are usually quite stable inside the language family. As travelers of Europe, we visit countries, we know a bit how to start to orienteer some, maybe the numbers, the way to say thank you or hello. And then you go to some countries and you have zero references to orient. This is how fascinating it can be, the linguistic diversity of the world. Think of Europe as in most countries, we speak languages that are related ultimately. But if you think of other regions of the world, like in South America, in some regions in Africa or in Asia, the normality is to have your neighbors that speak languages from different language families, and even to master more languages, to be multilingual, to normally speak languages from different language families. So, yeah, depending on where you travel, you will find different linguistic profiles.
It's really fascinating to me. Also, in Europe, as you say, quite a few languages are related. So you would be able to identify, is it Slavic? Is it, But you mentioned South America, and I know you did a lot of work in South America. You did a project on the Inca Empire and the spread of the Quechua languages. Would you like to tell us more about that?
One of the largest language families in South America is the Quechua one, at least spoken today by a very large number of people. It's spoken mostly in the central Andes of Peru, Bolivia, up to Ecuador, and a little bit in Colombia. And at the present time, we don't know when and how Quechua started to diversify. So there are different theories proposed. Some more in a more superficial way associate the spread of the Quechua language family with the moments of expansion of the Inca Empire. Because this is the main history that we know, because the Spanish colonizer spoke with the Inca when they arrived there, and they got their version of their story. And this is how far it goes if you stay superficial. But of course, the Inca are just one of the last elements of a very rich and complex history that has been going on in South America for thousands of years. So the idea is to sit together with archaeologists, linguists, geneticists, historians to evaluate the possible scenarios to explain when Quechua really started to spread and dominate these regions as the main language spoken then today in very vast regions. So different empires, and we call them empires, but different movements of cultures have been influencing the Altiplano, these highlands of the Andes, up to the lowlands of the Amazonia region. And it's interesting to disentangle the effect of this population movement also on the languages.
So, who speaks this language today?
The history of the Quechua language family is of 2,000 years.
Okay.
The history of the Indo-European language family, depending on different date and different reconstructions, might be 4,000 years up to 8,000 years old. So language families can stay for many thousand years, diversifying in sub-branches, other languages. What used to be defined maybe a dialect at some point gains a status of fully distinct language. So when does Quechua start in the chronology of the Andean highlands? We cannot fully pinpoint this point in time. Might be not too far back in time. The language today is still spoken because also of processes of revitalization and inclusion, where again, as I mentioned before, at this point, like when a language gets stigmatized, when an imposing language comes to a region, sometimes it will stop to exist. But the idea today that many people are doing in Peru, in Bolivia, is to keep the language and the identity that comes with it alive with a status of importance for the country.
Thanks for clarifying. I think we're all so interested in languages, but like, at least for myself, the knowledge is quite limited. So I didn't even know that these families were so old, like the Indo-European. I probably read it somewhere at some point, but it's good to refresh your memory here and there. So let's move on to our follower questions. The first one is, why do we speak so many different languages? And let me add a sub-question to that. How many are there? Is it around 7,000 or am I completely wrong?
Yeah, depends on how you count, but let's say it's a bunch of thousands of languages that we speak today. On this, we can agree. This is a very common question, and it's again very fundamental because everyone has a language, right? People call it the dialect sometimes, but it's always a language. A sign language is a language, so in principle, everyone has a language. How do they come in this range of diversity? Some languages are considered to be simple, and some are considered to be more complex. Some elements in a language require even more effort at a cognitive level to be assembled together. Languages range between different span of diversity of their features, and there is no like optimal way to have a language, otherwise we would just speak one language, right? So it's intrinsic in our history, in our identity. The diversity is pushed into many directions, and the outcome is all of these languages that we speak today.
Can genetics explain why certain ancient languages have disappeared over time?
This is a very good question, and I'd like to have actually a project about that. There's some chat that we are having with the linguists that indeed ask these questions. There are interesting cases focusing on Europe, for example. Etruscan is a language that used to be spoken in central Italy for a very long time. Is a non-Indo-European language. And then at some point people stopped speaking it, right? So some elements of these ancient languages that disappear might still be surviving today as a linguistic substrate in the languages that we speak in the same regions now. Genetics can tell us if there has been a discontinuity in time, if there has been like a migration of people replacing the people that were living there before, or if it has been an assimilation of existing lineages or ancestries of the region. And this can contribute to understand the degree of linguistic replacement in some region.
What's the most surprising thing you've found about language evolution through your research?
Surprising things. Some results from previous research. Like when we studied, for example, the genetic history of a region in the north of Peru that is called the Chachapoyas. And the people living there have been associated to a specific culture, a specific kind of pottery or architecture, and traditional history, like the chronicles collected by the early Spanish occupants of the region, told us that the region was completely emptied by the Inca occupation. The Inca were fighting them so much and they were resisting them so much that when the Inca finally got over that region, they wiped it out and they relocated everyone to other places of the Inca Empire, sort of punishing them for being so rebellious to their domain. We did a genetic study in the region also tracking this very particular type of language that is spoken there. And we find that the genetic ancestry of the people living today there is quite peculiar and doesn't really match the genetic ancestry of the rest of Peru, for example, for where we have data from. So it seems that the Inca were telling a story that was a little bit exaggerated, saying that they were wiping them off and replacing everyone. This is something that we've been discussing, and also some archaeologists would say that this reconstruction makes sense.
That's truly interesting. Another case of propaganda of an empire.
Yeah.
Okay, let's move on to our HEAS Quick Take segment. Just answer spontaneously, okay? Where are you from?
Italy?
That was easy. Winter or summer?
Summer?
You have beautiful summers in Italy, so which languages do you speak and which one would you still like to learn?
I speak Italian, I speak Spanish and English. I speak a horrible German that allowed me to go far until now, and even a bit of Swiss German that I shamefully mix with the German, creating my own version. I would like to learn better German, but I gave up. Now I live in Sardinia, and I would like to learn some Sardinian, which is not spoken a lot, but still spoken, and you can hear it on the street. And when you know a bit of the language in the place where you live, you are part of the culture a bit more.
That's interesting. How different is it from Italian, the Sardinian?
Sardinian is quite different from Italian. It's a language that traces its root quite back in the tree of the Romance languages, as we say. It has some archaic features that makes it look like Latin a bit in some cases.
The more you know.
It sounds really beautiful.
And can I please ask you to say one word in Swiss German for us?
Gipfeli. The croissant.
Cat or dog person?
Everything.
Same.
Even tardigrades. All the animals are fun.
If you could have any superpower, what would it be?
Traveling in time.
I can only imagine. Yeah, that would be fun, because then you would have probably all of the answers to your
I want to see what people were speaking before the Inca in those regions of Peru, and then finally we would know.
Finally.
But
I'd love to play fly on the wall as well. A hobby of yours?
Dancing tango.
Oh, love that. What do you collect?
Everything. I
Oh,
don't like Marie Kondo lifestyle. I collect nail polish bottles.
oh, I'd have a few of them at home as well.
I have hundreds.
Oh my God.
I like to do nail fancy patterns.
Oh, wow. But do you have like a shelf on the wall where they're all, like in nail salons where they put all the nail colors and
No,
color palettes?
there is a limit to dust keeping stuff, so I put them in a box.
Okay. If you weren't a scientist, what would you be?
Some job that has a lot of traveling involved. A secret agent, maybe.
That would be cool.
I watch too many movies.
Oh, but they're good. I like that genre as well. So, Chiara, we have another segment that is called Speakers Ask Speakers, where we ask our guests to leave a question for an upcoming speaker. And today you have a lovely one. And as you like traveling, maybe you will enjoy that one. That person is asking you what you were looking forward to doing when in Vienna. So, is there like a special food you would like to eat or try or something you wanted to see?
When in Vienna, people want to see a bit of Klimt and Schiele, usually. Lovely artist. I've been to Vienna already, and of course, the first thing I wanted to do was to see the museums with the paintings.
Yeah. Okay. And last but not least, where can people find you online?
I wanted to say Twitter, but nobody's in Twitter anymore.
I saw your website, which I like quite much.
I keep something on the website, and trying a bit of tweet some every now and then.
Okay, I'm gonna put both in the show notes below. Chiara, thank you so much for chatting with me today for the HEAS podcast. It was a pleasure.
Thanks. It's a pleasure. Thanks for organizing this.
Thank you so much. Hello again, Victoria here. If you enjoyed this episode or the podcast in general, please consider showing us your support by liking, sharing, and subscribing to our podcast on Spotify and YouTube. Your feedback and engagement mean the world to us and keep us going. Thank you for joining us on this journey and stay tuned for the next episode of the HEAS podcast.